The Human Disease Ontology presents Mining Disease Information via Imports, Connecting Disease-Related Information. So, what is an import? An import is simply the importation of another ontology, whether it be the entire ontology or a subset of terms. The Human Disease Ontology uses imports to provide a more in-depth description of human disease. Many ontologies rely on imports, and just a few examples of such ontologies include the following. The Human Disease Ontology imports and utilizes 11 ontologies, allowing for different views of diseases through these imports. Imports reveal connections between diseases through the integration of biological information about diseases, including their symptoms, phenotypes, and genetic or environmental drivers. Links to these ontologies can be found below in the video description. What is an axiom? Axioms and ontologies can be thought of as logical definitions. Axioms are logical assertions that define relationships and allow for viewing of inferred relationships to build more complex multiparental disease-to-disease relationships. Axioms allow for broader understanding of what the disease encompasses. Axioms allow for inferred multi-parentage of terms in the doid.al and doid.obo files, which behind the scenes connect diseases by shared attributes. Axioms are built in three parts. Part 1. An ontology relation term, such as located in, is used to describe the disease. In Part 2, property restrictions, such as sum, min, max, and only are used. And in Part 3, an import term is used to build the relationship. The use of axioms to infer relationships allows us to view diseases on the basis of their anatomical location, their cell of origin, their mode of inheritance, their sequence structural variant type, symptom, phenotype, or even environmental exposure. Let's look at an axiom example using spinal disease. To define all diseases located in the spine, we first define an equivalent to axiom, or EQ statement, for spinal disease, which would be disease and located in some vertebral column, where vertebral column is a term in the Uberon ontology import. Then, to specify diseases that are also located in the vertebral column, we add the subclass of statement located in some vertebral column, which exactly matches the EQ statement to these diseases. On the Human Disease Ontology website, simply click on the highlighted words in the EQ statement to view other diseases located in the same anatomical location. For example, scoliosis. Note the subclass of statement, which defines scoliosis as a subclass of spinal disease via the logical axiom. Let's look at some examples of human disease ontology imports and their use. Axioms allow us to infer relationships where there is an allergic response to a disease that is not classified as an allergy or as autoimmune or hypertension. The relation term has symptom from the symptom ontology import can be used to infer and relate a number of diseases that are not asserted as allergic diseases. For example, the disease term pleural tuberculosis has the subclass of axiom has symptom, some allergic reaction. A few other examples include allergic urticaria and allergic contact dermatitis. Axioms allow us to infer relationships for additional diseases that involve an immune response that are not classified as an allergic disease. The relation term has phenotype can be used to infer and relate a number of diseases that stem from the human phenotype ontology import. For example, the disease term B-cell deficiency has the subclass of axiom, disease, and has phenotype, some abnormality of immune system physiology. A few other examples include dendritic cell deficiency and T-cell deficiency. Links and references to the resources mentioned in this video are available below in the video notes. Hopefully, you found our description of how the Human Disease Ontology uses imports to be helpful. If so, please like and subscribe. This video was brought to you by Lynn Schrimmel and the Human Disease Ontology Project. Funding for this project is provided by the National Institutes of Health National Human Genome Research Institute, grant number U41 HG008 735-01A1.